diploid fish, and not many people have done this. So I wanted to show you how the process works. So this is actually pictures, mixed pictures from the last couple of years of us going out this time of year. In fact, I just got back a week ago from spawning fish out in British Columbia at this fish farm. So you can see that they bring the fish into the tanks in the hatchery in fresh water to adapt them to fresh water. And basically they're starting mature, and being in the fresh water accelerates the maturation process. So you can see Elliot, the farm manager, fishing a fish out of a tank there. You can see some of the fish we fished out. And as I said, these fish, you can see they're missing bits of skin. Sometimes their eyes are popping out. Their tails, you can see the skin's gone and there's really just the bones hanging out there. Somehow they swim up rivers anyway, but these fish are starting to be in rough shape, but they're ready to spawn. And of course, you fish out the males. All of these fish are going to die after they spawn anyway, so we euthanize them before we start. But in the case of males, you actually squeeze the milt or the sperm out of them while they're alive. So you can see there is a postdoc from my lab, Aaron, catching the milt as it's being squeezed out by one of the hatchery people. You put the milt into a little paper cup, then you go and collect females. So you fish out the females. The female fish, again, you have to euthanize them. You don't want any blood in the way because the blood contains fresh water, and that helps activate the spawning process. If it drips on the eggs or the sperm, it will activate the spawning process before you're ready. So you can see me there in the lower picture, drying the fish off very carefully after we've collected them and cut the gills to let the blood drain out of them for a little while. So once you've got the females, then you have to get the eggs out of them. As I said, they're already euthanized. So this is John Heath holding the fish up in the upper left there with Jane Drown, who's someone who really likes to help out with spawning. And this is how you collect eggs. I'm going to show you a video here. Oop. Sorry. I turned the volume down. So you can see the fish is dead, so you can just hold the fish upright and slice open the belly. And if the fish is ripe like this one is, mostly, the eggs will just drop out into a bucket, and you can collect a few out of there and put them in a bucket. And then hopefully you can see, as Jane here is on the right, you've got a nice bucket full of eggs that you can use for spawning. Now, the next step is, of course, to mix the sperm and the eggs. So you can see we mix the sperm and the eggs. In our case, we were doing 3x3 crosses. So each 3 males got crossed equally with 3 females. So each of those 3 styrofoam containers would contain eggs from a single female, and then we'd mix the milt or the sperm from 3 males in there, and then we do that 3 times. You then stir. You can see there's a very interesting implement, your finger, for stirring the milt and the sperm. You wait about a minute for the eggs to fertilize, and then you add some fresh water. And as I said, the fresh water sort of helps finalize the process. The micropore on the egg closes and there's no more fertilization, and it actually hardens off the eggs. The eggs get a little bit more firm on the outside and less soft, and they're ready to go. Now, for diploid fish, we would normally just put these away in the trays that I'll show you in a second. For triploid fish, you have to tripletize them. And there's a couple of ways of doing that. One is to heat shock them, and the other one is to put them under pressure, which is the version that we do at Yellow Island. So you put the eggs that you want to tripletize in these baskets, and then you can see they've got this pressure machine here. It's essentially like a French press, if you look in the middle. So you can put the baskets in that steel cylinder that's in the center. Then you crank down the piston until you get up to 9,600 PSI, which is, I still think, I'm amazed that the eggs can stand that pressure. And you keep them there for about 5 minutes. And you have to do this pretty much exactly at 30 minutes after you started the fertilization process. So you're retaining the second polar body. And then you take them out. And when you're done tripletizing the fish or dipletizing, then you put the fish into Heath trays. So you can see they go into little compartments in these trays. And then you can see the stack there on the left, and fresh water drips down through almost like a stream. And then the fish are ready to, they incubate and become eyed eggs and then swim up. And then eventually you've got to take them out of there and put them in tanks and feed them in the freshwater stage up until so about June of the next year. So we spawned them now. They will swim up maybe February, put them in tanks, feed them in fresh water till June to September, and then put them out in net pens. So for those of you who are master students, you can see that this kind of project is not really amenable to fast graduate studies because you have to take over a year just to get the fish into net pens. And if you want to study them in fresh water, you really want to leave them another winter. So it's 2 years before you really can access the fish in the saltwater phase, right? So we've done this a couple of times now on this project, trying to develop triploid fish. I want to talk about the first experiment we did. So we spawned fish in the fall of 2018. And that year we produced about 3x3 crosses, 6 3x3 crosses, and we got about 12,000 fish. And then we wanted to do a big common disease trial where we took lots of fish, exposed them to a Vibrio bacteria, and then we'd have lots of samples for everybody on the project to use for their various projects. So you can see here we've got 15 of the families that we chose. So from the 3x3 crosses, the ones that worked the best, we felt. We had about 30 fish each from each family, so about 450 fish per trough. And we have replicate, triploid, and diploid tanks that are going to be fed a probiotic feed, and regular replicate, triploid, and diploid tanks that are fed regular feed. And we've got some controls on the end there that are just fish that are not going to be subject to the disease trial, but we just put them there to make sure that they live through the whole process and there's no problem with the tanks of the water to cause mortalities. We're really interested in comparing the triploids and diploids though. But you can see there's about 450 fish per trough. So this is about 4,000 fish in this experiment. And we exposed them to a Vibrio. I should say that the first probiotic we used was Jameson. So it's a commercial probiotic you can get off the shelf at Costco. We'd done some preliminary trials to show that in very small fish, like 10 to 20 grams, if you fed them the probiotic, there was an enhancement in growth. So that was the one we decided to use for this year's experiment. And as it turned out, it was not perhaps the best one, but we'll get to that. So again, we have a strain of Vibrio angularum that we use. Over the years, we've tried a couple of strains, but this one's very good. It usually gives us quite good mortality. Yellow Island, when they set up, they just let the fish die from diseases, whatever they was around, and then took the survivors and bred them, so that their stock is actually quite Vibrio resistant. Notwithstanding the fact that also their stock go out to sea, and every summer there's a Vibrio outbreak. So getting a strain of Vibrio that was powerful enough to kill them was a bit of a challenge. But we did, in fact, find this strain. So you can see the control fish at the upper left there, and then an infected fish right beside it. You can see the difference with the lesions under the skin. You can see the blood at the bottom of the gills there, and you can see the vent is sort of starting to bleed as well in the bottom. So it's a Vibrio that works quite well, and we can quite clearly see the symptoms. So how did it work? Well, this is just the survival after the Vibrio challenge. You can see there's 20 days post-infection that we monitored, and you inject them, and it takes 4 or 5 days for the mortality to start happening, but you can see that it is starting to happen. And if you compare the groups, what you'll find is that the probiotic groups actually did worse than the regular feed this year. And again, it's one of those vagaries of trying to do an experiment over a year. If it doesn't work, you've spent a year finding out it doesn't really work very well. This particular probiotic didn't work terrifically well. And we think it's because maybe the probiotic wasn't very good. And part of what we found out was the way that we're coating the feed with the probiotic is we dip the feed in probiotic, and then we spray alginate and calcium chloride on to put a hard casing around it. But the alginate sometimes doesn't help. And also, I just don't know if it was a very, very good probiotic. We fed them for a long time, and perhaps a probiotic has to be fed in just a short burst. So that year, we didn't really get a lot of improvement with the probiotic. And we've got lots of samples from this, and there's lots of things going on that I'll tell you about some of the other projects in the end that we're working on. But one interesting thing was we thought that it wasn't working very well in the freshwater, but we took a subset of these fish and put them out to sea in net pens. And just this year, we did a trial with them with Vibrio to see what happened. And it turns out that if you feed them in salt water, it actually reverses the effect, and the probiotic actually has a really good effect. So I know it's maybe hard to see the legend down below, but here's 10 days post inoculation with Vibrio angularum with the fish after they've been in salt water for over a winter, and you can see that the The gray there is probiotic fed diploids, and they had 100% survival. And then the probiotic fed triploids, the yellow line below that, actually had 97% survival. I will apologize. This graph starts at 80. We had to do that to accentuate the difference. It doesn't start at 0. So it's perhaps not as dramatic as you might think, but it is indeed an improvement. And then the next two groups were fish that were fed regular feed, but switched to probiotic feed when they went to the net pens. And then below that is all the groups that basically in the end were fed regular feed. So with the probiotic then regular feed triploids being the worst at 82% survival. So perhaps this probiotic is having an effect, but only in the saltwater phase of fish. And we've got, again, lots of samples of gills and spleens, and we're going to start digging into the hows and whys of the effect that the probiotic had on immune response in the upcoming year. And I do want to point out that the triploidization process is different for every family of fish. This is why we do lots of crosses. And I think this is why, in my view, epigenetics is one of the things that really needs to be looked at in fish immunology, because every family had a very different response. And you can see a big variety there. The 3 arrows are 3 families we're going to look at that had a not so good survival rate within the larger crosses. And the three green arrows are families that had a much better survival rate. But if you just compare the top one, family 13, you can see the percent survival. You can see the replicates there for triploids, on regular or probiotic diet was like 35, 38%. But for family 27, it was 51 and 67%. And you can see similar improvements in the probiotics. So these fish, the triploidization process affects the families different, the genetics different. And perhaps there's, we can find families that will respond better to triploidization and spawn them for the future. But I'd like to understand why those differences are there. But you can see we have a good chance of doing some studies on immune gene regulation and immune other factors like that to see if we can see a difference between those two groups. Now, I was talking about MHC genes and how they're important, and I want to know the MHC gene structure or allele content of these animals. This is a project that Maureen Latimer from my lab is taking on. And so here's, again, some of the crosses. We did 6 crosses, as I said, with 3x3s, 3 females crossed with 3 males. And we picked some of these crosses to genotype to look for MHC alleles. Now, the way that you genotype is, you can see this is a picture of the proteins, and you can see both of the proteins, both class 1 and class 2, sit on the membrane, but at the distal end at the top there's a peptide binding cleft. That's the part where there's a lot of diversity in the genetics. That's why there's thousands of alleles in the human population. Those differences are all in that peptide binding cleft region of the protein. So what you can do is you can PCR amplify that region of the gene that encodes that from each individual and look at the differences in the nucleotide sequence and see what the differences in the protein will be. And that will translate to different efficiencies binding to peptides and probably different responses to pathogens or survival. So we did this just by hand at first. So you can see we amplified just the peptide binding region of, this is MHC class 2 beta, from a different set of parents and offspring. You take those PCR products and you clone them and put them into a plasmid and then transfect some bacteria. Then you pick the white colonies and you send them off for sequencing. And again, you get back sequences that look like this. And these are actually different alleles. I didn't align them for you, but you can see we did find 3 or 4 different alleles in the fish we were examining, just the small sample size we had. Now, we named these alleles, so you can see here just the set of parents up above. We have the 4 alleles A, B, C, D in their distribution, and then the offspring. And excuse me, just a sec. You can see that we can trace from the parents what the genotype of the offspring should be. And this is just a small sample set, but you can see already that this offspring 27A has alleles A, B, and C. When it came from parent M9 that had C and parent F9 that had A and B, it seems to have all 3 alleles from its parents. So that's interesting, because that would seem to say that it's retained an extra allele in the triploidization process. These are all triploid offspring. But we have to do this at a much larger scale. And what we've done is we did this at first just by hand to make sure that we're seeing the right alleles and know what we're doing. And then we've designed next gen primers, and we've taken 270 offspring and the parents, and we've done it all with next gen sequencing. And Maureen is starting to analyze that now, but she's kind of locked out of the lab and the computing power that she needs to do it, but we're working our way through it. But it is interesting that we might see some of these triploid fish retaining extra alleles that they should have perhaps gotten rid of during the developmental process. Now, we wanted to try different probiotics because the Jamesons didn't seem to work too well. So this year, we took some fish that we spawned last November in 2019. We did 9 3x3 crosses, and we took some of the fish and tried different probiotics. And so again, we've got a setup where we've got the control. In this case, we set up a control where the feed was just coated with the sodium alginate. It was something we didn't do the first year, just to make sure that our suspicion that the alginate is having a negative effect is tested. And then we tried these various formulations of probiotics called Seed 14, Seed 19, RC14 GR1, and Pediococcus. And again, 30 fish from each families. We only took 5 families, 150 fish, because we traveled to BC in the middle of a COVID lockdown, and the number of people we could send there was limited. So the number of experiments we could do was limited. And you can see, again, this graph starts at 55%. So again, we're exaggerating the differences a little just to show you. But you can see what happened this year is that the fish with the regular feed or with the sodium alginate are the two bottom lines, the blue and orange, and they had about 60% survival. But the probiotics, particularly the Seed 14 and the Pediococcus, actually enhanced survival up to about 65%. So it seems like we had the opposite effect, and we've actually got some probiotics that enhance survival in the triploid fish. And so maybe it's helping ameliorate some of the immunosuppression. Now, again, because of space and time constraints, we didn't test the diploid fish this year. So we're not sure how close that is to the diploid survival, but it's an improvement. And I think in general, based on previous experiments, the triploid fish did 10 to 15% worse than the diploid fish. So if we can even get 5% survival back, then we're improving the survival for the farmers. So again, COVID lockdowns prevented us from analyzing a lot of this stuff, but Maureen is going to analyze the MHC sequencing products of all parents and offspring, those 270 individuals, by next gen sequencing. We are going to take some of the triploid and diploid fish and actually make back clones and sequence the MHC region to try and see what the genome structure is and if there's genome content variability like we see in zebrafish and salmon. Manuel de Soto, who some of you may know, who's working on this, is going to do a lot of work on measuring the immune responses by PCR and ELISA to see if you can see differences between diploids and triploids in responses to the probiotics. And we also bought a little portable flow cytometer so we can actually look at the number of T cells, CD4 and CD3 positive T cells, in triploid and diploid fish, and see if that theory that the triploid fish have lower T cell counts is actually true and holds up and matches the number of alleles of MHC that we see in the other experiments. We also have lots of other things going on. As I said, we try to do experiments with lots of fish so that the other labs can have fish to work on. Paul Craig, whose lab is here in Waterloo, has a student called Ivan Kodonik who's going to work on epigenetics and look for miRNAs that will help perhaps be affected and regulate, and we'll try and look at some methylation as well. The Semyonuk lab in Windsor is actually looking at behavioral differences because the triploid fish tend to behave differently than the diploid fish. They're not as aggressive. They hide at the bottom of the tanks. You throw feed in, they don't come up and get it. And we're trying to look at behavioral differences that might lead to immunocompromised conditions or might enhance disease susceptibility. And again, if the probiotics somehow ameliorate that. The lab that we used to work on the probiotics is Gregor Reed's lab from Western. He has a student, Luana Langlois, who's working on looking at the effectivity of the probiotics by looking at gut barrier genes. And Dan Heath in Windsor has a student, Edel Bai, who's going to work on the gut microbiome and look to see if the probiotics actually change the gut microbiome and consequently change the immune response in the fish. So that's the triploid project. Again, here's many of the people involved at the upper left there. You can see myself, Paul Craig, Ivan is the student, the tall student with the hat, Maureen and Manuel standing beside me. And then there's other people from my lab there who are also involved in experiments there. And that's funded by NSERC. It's an NSERC strategic grant and my Canada Research Chair. Now, for the East Coast, as I said, the focus is very different. They're not much more worried about jobs and industry than they are about the environment. Not to say that they don't care about the environment, but the focus is different. It's about, protecting industry. And the industry on the East Coast is threatened by climate change. So if you look at this projection, you can see the sea surface temperatures in 2011, and the sea surface temperature around Newfoundland and Nova Scotia is currently hits a peak in the summer of about 16 degrees. But by 2035, it's predicted to hit 19 to 20 degrees. There could be a 3 to 4 degree increase in the temperature. And for those of you who know anything about salmonids, you'll know that 19 to 20 degrees is probably not a good temperature for them. It's when they start to get stressed. And we have a collaborator in Australia who works on salmon aquaculture in Tasmania, where the water is the peak water summer temperature can already get up to 19 degrees. And there they don't even go on the net pens to feed the fish for a few weeks at the high temperature season, because just feeding them will stress them enough and they will die. So the project here is called MIXA. It's funded by ACOA and AIF. And the main project is to breed strains of fish that are temperature resistant, that will handle the stress and can handle diseases at high temperatures. I'll talk a little bit more about the project later on, as I said. My part of it is to look at protein level assays for key immune or stress markers. Because if I worked on human immunology, and I have been lately, I don't really like it, but I'm doing it. I can order antibodies to immune markers from 3 or 4 different companies. I can phone a company, have an antibody to an MHC or an interleukin-1 beta cytokine or whatever I need, within a couple of days. For fish, those tools just don't exist. And so my project has been to develop assays to measure the protein level, interleukin-1 beta or other stress markers in the fish, because that's a much more functional level. And again, the whole idea is for this project, we would use them in assessing the families that we have to see who's got better or different immune responses at high temperatures and who will be able to survive in the face of climate change, right? It's again about preserving the industry and making sure that it's there for the future. So why is looking at the protein level important? Well, the thing about the immune system is that if any of you have taken an immunology course, it's complicated. And of course, genes, you can see the gene in the middle of the big blue cell there. Genes get transcribed into transcripts and messenger RNA, and you can always measure that, but that's not always a reflection of the protein level of the product that the gene is actually expressing. If you look at our messenger RNA, it has to travel to the cytoplasm and be translated by a ribosome into a protein. But in the case of the immune system, that protein has to be transported to the cell surface, which is also a process that can be regulated very tightly. And depending on glycosylation, you can change the level of proteins on the surface to match the environment. Or if it's a cytokine, for example, a small soluble messenger that's going to affect another cell, it can get secreted. And the thing about a lot of these molecules is they're secreted in inactive form. In my immunology class, I tell people that the immune system is the most paranoid system around. It wants to activate in response to pathogens, but it's also very worried about self-reactivity and autoimmunity and reacting to self. So it's regulated very tightly. So this cytokine protein that's secreted into the extracellular environment perhaps isn't even in its active form, right? And in order to get it active, it may happen outside, it may happen inside. It has to be cleaved by an enzyme into a proteolytic fragment. And then that fragment is the active form that will then go on and bind to a receptor on the surface of another cell and have an effect. So lots of things in the immune system happen at the protein level or at the cellular level. It's cell-cell interactions. So you really need to look at these secreted factors, these proteins and or cell surface proteins themselves, and not just look at the transcripts to get a full picture of what's happening. So these assays are very, very important. And that's why in human immunology, there's lots of these assays available. So as I said, we're going to develop protein level assays for key immune or stress markers. Um, Again, it would be a good diagnostic assay for immune or stress responses in salmonids. It's a good tool for assessing immune responses to see if vaccines are working. There's lots of things you can do with these things. The type of assay we want to use is ELISA, and I'm going to talk about ELISA today. It's a way of measuring protein content. ELISpot, which is a variant on the assay I'm going to talk about, but looks at the number of cells producing the cytokine. And again, that's important when immune responses happen. The number of cells that are active and secreting cytokines increases. So if you can measure that increase in the cell number, you can know something about the immune response. Or, eventually, if we get down to a very good marker, we could do a dipstick test, a side-loaded ELISA, like the diabetes test lately, or the pregnancy test, or some other multiplex ELISA to look at multiple cytokines at once. So how will this be achieved? Well, one of the partners of the MIXA project is Somru. They're a PEI company that makes antibodies in chickens. And we're going to work with Somru, and we've been working with another company called Cedar Lane to develop 20 recombinant proteins from immune or stress markers from fish that we can make assays for. And in years 1 and 2, we used known markers of stress or immune response, things that we figured were important. And then later on, when we started doing experiments that have now been conducted on stressing fish and doing disease trials, we looked at RNA-seq and we picked markers from those things, those experiments, to inform us about what we wanted to make assays to next, right? So as I said, we're going to develop quantitative ELISAs and ELISpot. And one of the other things about the immune system is that these proteins are actually good for stimulating other cells. So fish immunology has come a long way in the last little while, and we now have cell lines, but those cell lines in human studies need to be stimulated with various factors. If we can make folded active recombinant protein forms of these cytokines, we can use those for tests to understand immune function again. And actually, we're coming quite far with that. I'm not going to talk about it, but Aaron Furnett in my lab has been working with some engineers who are now expressing very high quantities of folded active protein in human epithelial kidney cells, and we're going to start expressing them in fish cells to make sure they're folded absolutely correctly. So today I just want to talk about interleukin-1 beta. It's the one we're just about to write a paper on. It's the one we're closest to finishing on. Interleukin-1 beta is a pro-inflammatory cytokine. So it's a small soluble messenger that's secreted by some cells in the immune system to activate other cells, and it's pro-inflammatory. It starts the response to pathogens. It starts in mammals, it starts a fever. In fish and in mammals, it causes inflammation and an influx of cells into the area. So it's a mediator of the response to microbial invasion. As I said, inflammation, it causes swelling and an influx of cells. It also responds to tissue injury, and it upregulates the expression of other genes. It's one of the earlier genes in the cascade of cytokine expression, and it does upregulate other cytokines that have knock-on or further down effects. It activates acute phase proteins, which are things like clotting proteins and complement in your blood that help fight off the pathogen until your antibodies and your adaptive immune system can get involved, and cell adhesion molecules to facilitate those cell-cell contacts that are super important for the immune response. So as I said, we developed a sandwich ELISA. It feels really not satisfying to present this in one slide because this took us years of work. But we have developed an ELISA. So an ELISA is basically a 96 well plate based assay. So you can see in the figure on your left, there's a little, bracket at the bottom that is supposed to be a well of a 96 well plate. And of course, this isn't to scale. We would coat antibodies on there. So we would take an antibody that we've developed to interleukin-1 beta, and we coat it on the plate, right? So this is a sandwich ELISA because we're going to sandwich the cytokine with 2 antibodies, but it's a very quantitative method. So you've got this plate pre-activated with an antibody to your target, interleukin-1 beta. You can then take the blood or serum sample from your fish to see if there's any interleukin-1 there, and you let it float in there, and then the antibody, if it's the capture antibody, will grab onto it if it's present, if the fish has an active immune response, right? You then wash away whatever's left and use a detection antibody. So in many mammalian systems, the detection antibody itself will be linked to an enzyme that you can use to detect the presence or absence of that antibody. But in our system, there aren't very many pre-made antibodies like that, and we had to make this rabbit antibody ourselves. So we use a rabbit anti-interleukin beta to detect, and then we add secondary antibodies. And in fact, we add biotin and streptavidin beyond that, because each of those steps, adding antibodies, the secondary antibodies, the goat anti-rabbit, and the biotin, amplifies the signal. These proteins like interleukin beta are present at basal levels in the 6 to 10 to 12 picogram per ml range, and can go up to 800 or 1,000 or more when they're activated. So to get down to the basal level, we have to amplify the signal a little bit. And we found a way to do that with very little background. That's what took us several years. So you can see here on the graph on the right our range of detection. And you can see we can quite readily detect from 6.25 to 800 picograms per ml of cytokine in the blood, which is quite sensitive. And we're able to deploy this quite regularly now. So the first thing we want to know is, does this assay work? Does it detect interleukin beta when it should be there and not have any spurious or false negatives? So the first experiment we did was with RTS11. So that's a macrophage monocyte cell line from rainbow trout. Just a sec. And it should be a potent producer of interleukin beta when activated, right? So we used, again, heat-killed Vibrio. Vibrio is a good model for us in our lab as a pathogen. And basically, we set up a 6-well culture plate. Of course, we replicated this, but we put killed Vibrio in 3 of the wells and control RTS11 in the other. We added just PBS instead of the Vibrio and PBS, and we incubated it for a little while. Excuse me, I'm gonna have to cough again. All right, and then we sampled the cells at 1, 4, 12, 24, and 96 hours post-stimulation. Okay, and basically what we're looking for is to detect intercellular and secreted interleukin beta. So first of all, you want to check that you're definitely stimulating the cells. So the first thing you do is look for altered morphology. I don't know if you can see that in your slide, but in the controls, the cells are all very round and they're non-adherent, they're floating around. But in the Vibrio treated cells, in the next panel, you can see that a lot of them are elongated and they've adhered to the plate, and they've got attachments there. They're definitely activated. They're becoming macrophages and adhering. And if you look at the PCR results for interleukin beta, you can see that interleukin beta messenger RNA appears about 4 hours, peaks about 12 hours, and then diminishes out to 26 hours. And the control wells, there's no detectable interleukin beta message. How does that compare to our ELISA? Well, you can see this is the fold change. So at 0 and 1 hour, that's 1 fold. That's essentially the basal level that you'd have of interleukin beta. And you can see that the level increases, particularly in the Vibrio infected ones. You get a significant difference to control, which doesn't really change from the 1 fold or the basal level that's there. And the peak is at about 24 hours, about 12 hours after the peak of the messenger RNA, which makes sense because the messenger RNA has to make the protein. So protein expression lags, but protein expression is definitely diminishing again by 96 hours. So it looks exactly like it should. We've got messenger RNA and we're detecting protein where we should detect it in the stimulated cells. And you can see if you do a Western blot, you've got the 0-hour control lane there and the 24-hour control lane, and there's no real detectable interleukin beta protein there. So this is basically a way of looking at the protein where you lyse the cells and put the lysate onto a membrane and then probe the membrane with your antibody. But this is basically protein that's inside the cells because we're lysing the cells. So you can see the 0-hour control and 24-hour control. There's no interleukin beta there. In the Vibrio treated cells, there is a band, a single band you can see that's approximately the same size as our recombinant protein that we produced in the lab. And again, there's a slight difference there because there may be differences in glycosylation in the native state. And also our recombinant was made, I think, from a slightly truncated version of the protein. But there's only a single band there, and it's roughly the size that it should be to be interleukin beta. So we're pretty convinced that our assay is working very well, at least for cell lines. Now, to show that this works also in cells extracted ex vivo from the animals, we did the same thing with trout splenocytes. So We again took the splenocytes out, treated them with killed Vibrio or had controls, and sampled them again at the same time points, 1, 4, 12, 24, and 96 hours. It's the same experiment, just with primary cells. And again, we're looking for intracellular and secreted interleukin beta. Again, if you look at the qPCR, you get pretty much the same pattern of stimulation, with the peak of the messenger RNA happening about 12 hours after stimulation in the wells. And if you look at the ELISA, you can see that it's actually a little bit more of a delayed immune response. So this isn't quite as pure population of cells as the RTS11. There's probably mixed lymphocytes in there, but you can see that we are getting an increase in the interleukin beta we detect at 24 and at 96 hours. So, and again, it may be diminishing over a longer period of time, but definitely we're detecting it where it should be. And again, if you look at a Western blot, the 0 hour and control lanes have no interleukin beta. We do get a band the same size as we saw in the RTS11, and we get this foggy band down below. And that is because interleukin beta is one of those proteins that needs to be cleaved to be active. And we think we may be beginning to see the active form of the protein with that fuzzy thing there, but I'll get to how we're going to figure that out in a second. Okay, so finally, you want to do it in whole animals. We've done this. Fabio Zanuzzo, who I can see is watching because I see his glamour shot there, did an experiment with stressing the fish with swimming. But we've also, this was one of the first experiments we did where we did heat stress. So, and this was done in Kurt Gamper's lab right there at the Ocean Science Center. So we took steelhead trout, we stressed them. So slowly over time, 2 degrees C an hour, from 10 degrees up to 24 degrees, and control fish were kept at 10 degrees. And we sampled them all the way through to look for cortisols. And you can see, I actually don't have the cortisol here, but the cortisols actually spike up and then come back down to show that they are being stressed. But again, if you look at our interleukin-1 PCR, we didn't sample between 10 and 16 degrees, but you can see the messenger RNA peaks at about 16, 18 degrees and then diminishes. And then once you start hitting 20 degrees, the level of the messenger RNA for interleukin-1 beta drops off precipitously. And there is a link between stress and immune responses, and it's quite well.
Okay.
Hello?
Okay.
I think you need to mute your microphone. So when the cortisol actually spikes, it diminishes interleukin-1 beta expression. And so what we think we're seeing here is the suppression of the immune system by the stress response as the interleukin-1 beta drops off at the high temperatures there. And again, if you look at our interleukin beta sandwich ELISA, the level of the protein, you can see it also peaks about 16 degrees and then diminishes down and also disappears once you start getting to the higher temperatures, 20, 22 degrees, with a little bit of a lag from the disappearance of the messenger RNA. And again, if you look at a Western blot with the cells, you can see that in the serum, you can see that we're actually seeing a band about the right size. In the middle there, you can see at 10 degrees, you're getting a band about the size we saw in the cells. And we're also getting that lower band that looks like it's the processed form of interleukin beta in the 16 and 24 degree fish. And again, compared to the recombinant protein, right? And that recombinant protein that we're showing there is we're showing you the mature or the processed form and the precursor or the pre-proteolytic form. And you can see those bands correspond roughly to the two versions that you see. So we think we're seeing interleukin beta quite well. Now I've got there we go. Now, again, I'm going to put some of these blots back up to show you the different versions of the proteins that we see. And this is one of the problems we have. We have an antibody and we see a protein on a gel, and it's a single band in some cases. It looks beautiful, but is it really what we're looking for? Because again, it's a slightly different size than our recombinant protein, and there is differences in protein processing. So what we're going to do is we're going to N-terminal sequence the protein. We're going to cut out the bands and send it off. And that, again, it's not as simple as it looks. We tried a couple of different partners to sequence the protein and haven't really got really clear answers. But we now have a partner that will sequence these for us. So we're cutting up these bands. And we're going to send them, and we're going to N-terminal sequence to be sure that we're looking at interleukin beta. And in the case of the proteolytic cleave form, we're going to look to see if the N-terminal sequencing gives us the sequence in the protein right after the proteolytic cleavage site that's predicted to make sure that it is in fact the cleave form of the protein. So what can we conclude from this version of this part of the talk? That we have a quantitative ELISA capable of detecting interleukin beta at at least 6 picograms per ml during in vivo, ex vivo, and that should be in vitro, one of those. In vitro, ex vivo, and in vivo stimulation. It works very well for all of those applications. The validation of the interleukin beta sandwich ELISA is supported by Western blot analysis, and the gene conscription patterns are consistent with it being interleukin beta as well. So we're just preparing a publication on this, and then we're going to actually release this assay. We're going to give it to Cedar Lane to market. So anybody who wants to use this assay can use it for their experiments as well. And again, we're going to work through the other 20 proteins on the list. TNF alpha is next, but there's some stress proteins there that we're going to work on to develop ELISAs and assays for those. So for this part, I'd like to thank, this is the MIXA project. So it involves all these partners down at the bottom. You can see here, Memorial, Prince Edward Island, Guelph, University of Waterloo, Elanco, Huntsman, Center for Aquaculture Technologies, AquaBounty, Somru, and it's funded by AIF and NACOA. And the people who contributed to the work today from Memorial would be Devin Ramsey and Fabio D'Annunzio. And in Waterloo, we've got George Heath, Aaron Fournette, Tanya Rodriguez Ramos, and Mark Woe. With that, I'll take any questions you have. And again, there's my contact info if you want to contact me for any reason whatsoever. I'm going to stop sharing because I can't see the chat while I'm doing this. But
yep. So please, anyone that has any question, just free to unmute yourself.
I have a question.
Hola, Javier. ¿Cómo está?
I'm Brian. I have a question that took a lot of my attention, the utilization of probiotics, in marine or salty water. So you show that the probiotics basically have 100% of protection.
Yes.
And what take my attention is probiotics. My understanding is are administered orally.
Yes.
So basically, they colonize the intestinal gut and likely they modify the microbiota.
Yeah.
And you are challenging these animals with an IP injection.
Yeah.
That is a different route.
Yep.
You get some really big, high protection. This is better than vaccines.
Yes. Well, it's way better than vaccines. Now, the one thing I should say is that that experiment was a preliminary one. And the problem was because the triploid fish didn't survive well. And in fact, the probiotic fed fish didn't survive well through the first freshwater trial. The number of fish in that experiment was not big. The number of fish we had in some of those groups was in the tens.
Okay.
So it looks like it's great, but it's something we're going to have to repeat because I'd like to do it again with hundreds of fish because it looks good, but it just wasn't as big of a number of fish as I would like to.
I was wondering also if this probiotic has any Vibrio component. Because maybe they are acting as a really potent oral vaccine.
I don't think there's Vibrio. I'd have to look at the list. It's a mixture of, I think, 17 different, but it's a human-based. You buy it at Costco, it's based on humans. So I don't think there's Vibrios, and there's a lot of E. coli's and lactobacillus and those kind of things. Now, the other ones that we're using this year also feature mixes of similar things. They're just perhaps fewer. But there's so many moving parts in this, like coating the feed and making sure the probiotics active, because for humans you can just feed it to them and it goes into their mouth. For the fish, the feed pellet has to go in the water, and we looked at I've got a postdoc looking at how much of the probiotic actually will grow once you dip it in water, and that cuts it in half to start with, right? So I think there's a lot of things you've got to address with how the probiotic's working. But it seems to have an effect, but I'd just like to know what it is. But yeah, we'd basically given up on Jameson's. We tried a couple of other trials. It hadn't really worked as well as we wanted to. And then we just did that quick test and it looked like it did something. So now we're going to have to look at it again.
Thanks.
Someone has another question?
C here from I'm a PhD student working with Matt Rice. I enjoyed your talk, Dr. Dixon. One thing, I think the first project you're talking about with Pacific salmon, you mentioned about a great variety of MHC locus, and you're talking about 2 anchoring genes, and they are showing a variety of form of MHC. So I'm wondering, do you see any evidence of even different paralog gene of MHC in different genomic location rather than with that particular location?
Well, again, the mapping of MHCs in fish genomes is not hard and fast. That is the region that's believed to be the classical encoding region.
Okay.
But and the data was actually showing with the 3 and 2, and the 1 was from zebrafish.
Yeah, that's right, yeah.
And again, with fish, the genes have moved around a lot. So there are other regions that contain MHCs that are not that region. And so for zebrafish, I think it's a little easier because they're diploid and they've had less genome duplications. Mapping it in salmonids, we know that there's alternate locations. There's at least 2 different locations where the class 1s are. Some of them are linked to the antigen presentation genes, they're not. So I think we're going to have to, as I said, I want to dig into some back cloning and look to see what regions I can find that match it and look for variation. And I will look for those flanking genes in that region, but I suspect I'll find those flanking genes in at least 2 different chromosomal locations because salmonids are pseudotetraploid, right? So
that's right, yeah. So you're anticipating that even at a different location, the flanking gene will be similar.
Well, they should be at least duplicated in salmons. And I think that I've looked at some maps, some preliminary maps from the salmon genome, and I think there is 2 regions like that. But again, fish are so diverse, you just don't know what they've done with their genome. And you may find that they're elsewhere. But certainly for PCRing messenger RNA, you find only 1 or 2 or 3 alleles. So I'll use those as probes, and that'll be the genomic region I'll have to focus on.
I see. Sounds good. Thank you.
Do we still have time for another question?
So I have another question.
Sure.
But it is about the Vibrio strain that they use, Brian. So how frequent is vibriosis in salmon?
In the West Coast, where the farm is, every summer there's a spike in Vibrio. You can measure it in the water, and every summer there's an outbreak of Vibrio. Certainly on all the farms around there, the summer temperatures get up to the point where it's there. So, and that's part of the issue. As I said, the fish are naturally vaccinated against Vibrio because they're out in the water with it, the saltwater ones. So it took us a while to find a strain that would actually kill the fish beyond that. It's very common. It's probably the most common disease that they have. They have some issues with BKD out there. But for the Chinook salmon, they keep them at low density to stop it spreading. So you see a little bit of BKD around, but Vibrio is basically their main problem.
Thanks.
So respect to the same, have you ever done any assay like in Vibrio angular infection, but in juveniles? Because I'm wondering if it's any chance that they can get more like not susceptible, like they're more resistant to the Vibrio angularum infection, because you can see that where they're going to grow, like getting more adult fish, they're getting more susceptible to different kind of strains of pathogens. But I don't know if that is the case in this.
Well, when we do the freshwater trials, those are juveniles. Those are fish that we spawned the October before. They've never been to salt water. So the freshwater phase is essentially juvenile fish. They're about 50 grams, 30 to 50 grams. That then when they go to sea, that they'll grow up to be much bigger, and we can test them again. The thing is, they're not often exposed to Vibrio in fresh water. It's really a saltwater pathogen. So we're exposing them and killing them to something but we're trying to be consistent with the same disease throughout the life cycle. So, but I think there is evidence they become resistant. As I said, this fish farm, when they started in 1985, just let fish die. However, it was never really set up as a commercial operation, I will say that, but they let fish die, and then they took the survivors and spawned them. And so they've got a pretty disease resistant set of fish. The first strain of Vibrio we tried, I had a student, Leandro, he was from Argentina, and he infected the fish and with a huge dose and got like 20% mortality. And he got frustrated because he wanted more. So he infected them again and he got another 3%. So they're pretty resistant to most, as I say, most strains of Vibrio. It took us a while to find this one.
And in your test of the probiotic also, have you ever done any bath infection?
We
Like
haven't
just
done
the-
any bath infection, and we've debated that because as Javier pointed out, you're feeding the probiotic orally. So if you give them a bath infection, at least you're
It's
getting
going to be-
oral infection or mucosal infection in the gills. It's something we're still toying with. The problem with bath infection that I don't like is it's really hard to know if the fish got infected or if it's a consistent infection because, and then you have to go down the route of maybe excising gills or excising spleens and doing PCR and eliminating the ones that didn't get it. So we may try it and see if it works, but I'm not a big fan of bath vaccination.
Mhm. Okay, so someone have any other question? I think that we are done for today. So I would like to thank you, Dr. Brian. Thanks for this great talk. It was very good. And hopefully we can be in touch, and we are done for today.
Okay. I'm happy to answer questions, and one of these days I'm going to get back out there and we'll do more work. We will. So, take care.
Okay, goodbye.
Thank you. Bye.
Thank you, bye.
Thank you. Bye.